So welcome, folks, and it is great to have you here, and I'm going to make sure to speak enough that you can hear me. If you're in line for drinks, just please keep doing your thing. Before we get started with tonight's Suds and Science, I want you to imagine a forest in Borneo and this small withered bat that's been desperately flying around all night in drenching downpour in the middle of a rainforest. This small brown bat, smaller than my fist, has been desperately searching for calories all night long, dodging raindrops. It is soaked, this bat is hungry, calorically deficient, and looking for a safe place to spend the day as the sun begins to come up on the horizon. This bat sees a cavity, detects it with its echolocation through the huge drops of rain, aims towards it, thinking it's a safe place to move into, to hide out for the day. Recognizing the cavity is a little oddly shaped, but plunges in anyway, desperate to get out of the soaking rain, to warm itself back up, to try to find some respite for the day. The bat lunges into this cavity, turns itself upside down in a quick flip, desperately tries to grab onto the inside of this cavity with its feet, but begins to slip towards the bottom. This isn't your typical cavity. This is a pitcher plant in the Nepenthes genus. Now, like all pitcher plants, they are killers. They are carnivorous plants that eat animals for their calories. Most pitcher plants depend on insects, maybe a small frog occasionally, to fall into the bottom of the pitcher plant to be digested by those juices down there. But this is not your average pitcher plant in Borneo. This pitcher plant doesn't get its calories from frogs or small insects. It gets its calories from bats. And in 12 hours later, part of this bat will be dissolved at the bottom of this pitcher plant. Having thought to have found safe refuge, it is being slowly dissolved. But luckily, the part that's being dissolved is not the bat itself, but its nitrogen-rich droppings. This pitcher plant and the Hardwick's woolly bat in Borneo have this symbiotic relationship. The pitcher plant doesn't eat the bat, but it gains calories and nitrogen in these bog nitrogen-deficient soils from the droppings of the bat that fall in there while the bat roosts. In the jungle, many things will kill you, and it's awfully nice to find a friend every now and then. Tonight's Suds and Science topic is about unlikely allies, much like this bat and pitcher plant in the rainforest of Borneo. Before we get into that, I want to say a huge thank you for being here, and welcome back to Suds and Science, folks. All right. And thank you for all you guys up here, too. We will try to make a point of turn around and so you can see us as well. My name is Jason Hill, and I am the long-term, for almost a decade now, host for Suds and Science. I'm a quantitative ecologist with the Vermont Center for Eco Studies. We are a nonprofit right here in White River Junction. And Suds and Science is an event series that we bring to you every month from January through May. Each month, we bring you a different expert in their field. This is an opportunity to lean in, to ask questions and to engage with an expert in their field. There's no PowerPoint to hide behind. It's just a speaker, an expert in their topic, and you. I encourage you to lean in to ask questions throughout tonight. Please be respectful of the speaker's time and try to keep us on track with the subject. But otherwise than that, put your hand up, ask questions. I encourage you to get up and to grab another drink or something to eat throughout the talk tonight. Please don't feel that you're stuck in your seat. Restrooms are all gender restrooms are out front, and I'll be back at the end of the talk to say goodnight. But it is my great pleasure to introduce tonight's speaker to speak about mycorrhizal fungi and trees, Dr. Jordon Tourville from the Appalachian Mountain Club. Put on your hand. Just speak loudly. Yeah. Testing.
All right. Am I audible to everyone here? Perfect. All right. Get it closer here. I'll just hold on to it, actually. Okay. Am I audible? All right. Thank you very much, Jason, for the introduction. As he said, my name is Jordon Tourville. I am the terrestrial ecologist for the Appalachian Mountain Club. Do we have any members of the AMC in the audience? Point, make yourself known. Thank you, sir. That's right. There's another one, there's two. If you are not a member, please consider being one. It's a great organization. It's one of the oldest environmental nonprofits on the East Coast, formed in 1876, so we're coming up on 150 years. And if you're hiking in the White Mountains, please at some point say hi to me. You'll probably be seeing me. Again, thank you to Jason. What I love about Suds in Science is that it really humanizes the people behind the science that you see every day. It's not simply this dull, dry endeavor. There are people with actual lives behind the work that's being done out there in the world. And tonight, I just want to share with you my personal journey into the sciences as we delve into the world of the things growing beneath your feet. If you're not aware, and we'll get into this, most of the biodiversity that we encounter on land is below the soil, perfectly out of sight and out of mind most of the time. But I'm going to try to argue that it has an enormous impact on the world we live in, the forests that we recreate in, that we depend on. But before we get to that, I'm going to take a bit of a circuitous route to get to fungi. And I'm going to first talk to you about sort of my own kind of weird wandering path into this world. So I am from Vermont. I'm a 7th generation Vermonter. I grew up on a farm in South Hero in the islands. If anyone's from there, give me a shout out later on. For me, I didn't have the best home life in the world. So the way I found solace was being outside amongst the trees. And from a very early age, I had the habit of trying to name all the trees that I encountered out in the woods. I was very young, I didn't know what the actual, quote unquote, actual name should be, but I gave, unique names to trees with unique characteristics. And I asked myself, why does this particular tree that looks like this grow here and not here? Why does this plant grow here and not here? And I accidentally stumbled upon one of the major themes in ecology, and that is, why do things exist where they do? And for animals, that could be, why does this particular rodent, forage and subsist here and not somewhere else? For a tree, it could be, why does it grow in this particular soil type and not in this soil type? To just give you a little bit of background what I mean, there was this guy, French naturalist, Alexander von Humboldt. In 1805, he had this wacky idea that he would try to climb over 4,000 meters in elevation up the side of a mountain in Ecuador with thick wool attire, terrible footwear, not very well prepared by modern standards. He actually made it pretty far up. And while he was on this journey, he noticed several interesting things. One, that the forests that grew at really low elevations were very different than the ones that grew at really high elevations. There was this very clear pattern of the vegetation as you go up in elevation. He wrote these ideas down, and he actually was a pretty good illustrator, and he actually created nice illustrations of this pattern of vegetation across space. The same patterns might be very familiar to us now. Is anyone here a very avid hiker, or has ever hiked in the Green Mountains or in the White Mountains? Make yourself known. Most of you, right? What do you notice when you find yourself at the trailhead, when you first get to the mountain? Anyone? Yeah, what do you notice about the trees? Yeah, they're big, they're deciduous. Deciduous, for those who don't know, this means the leaves fall off and they come back seasonally. Right. As you go up in elevation, you climb for a few hours, depending on your speed. I don't judge. What do you see? What happens to the trees? They get smaller. They become evergreen. This pattern repeats itself over and over again. No matter what peak you find yourself. Woo! Hold on. I got this. This pattern repeats itself over and over again on many peaks across the region. And this is not an accident. The reason that we, as ecologists, ask ourselves this question of why things exist where they do, because there are very clear patterns that are defined by things that we can measure. So in the case of our mountains here, things like temperature, precipitation, these things change as one goes up in elevation. And so the plants respond in kind. Evergreen trees are more adapted to these more stressful environments higher up on a mountain. These temperate deciduous trees at lower elevation are more adapted to those particular conditions they find themselves in. Now, this provides people like me and other ecologists very interesting little study system. These same patterns that exist on the side of a mountain exist across hundreds of kilometers of latitude as you go from south to north. So the forests of, say, the southeast, very different than the ones you find in the northeast, very different than the ones you find further up in Canada. It exhibits the same kind of zonation, if you will, as you go up north, further north. What's useful for us as ecologists is that we can use mountains where this pattern exists on a very small spatial scale to study big patterns of vegetation change. Now, on our mountains, there are a lot of things that are changing right now. Can anyone name a few things right now that may be not so stable? Perhaps think changing climate. There are other things. What's that? Yeah, changing patterns of disease, pathogens that are coming in. Water, that's right. Yeah. Tree line. So what I study is part of a phenomenon I like to call global change. Climate change is one aspect of the way that humans are altering the environment and the natural world around us. Primarily, we see this through increasing temperatures, changing precipitation, but there's other things going on. Does anyone remember a phenomenon called acid rain that was pretty predominant a few decades ago? And one of the few success stories where we actually made an impact with policy. That's another example of how humans are kind of tweaking the biosphere in a way, changing patterns of pollution, the introduction of invasive species. All these things are ways that we're altering the biosphere, and they're all kind of subsumed in this one term called global change. On mountains, we can see the effects of these changes pretty clearly. We like to think of these as sort of biological indicators of change. When the environmental conditions change around these plants, they respond to those changes. Like all organisms, trees exist within defined climatic limits. There's a certain temperature above which they do not grow very well. The photosynthetic machinery of the plant itself kind of breaks down. That's true at the lower cold limit that they can survive as well. There's a certain amount of moisture that they acclimate to, so on. And so as things continue to change, where you find these plants, the ranges of these plants shift in turn. And a lot of people have suspected that as things continue to warm up, plants will migrate upslope, they'll migrate further north. Now, they're not like animals. They don't actually move. They're not like Ents from Lord of the Rings. They can't get up and walk away. So the way they get around is by migration of generational movement, if you will. They disperse seeds somewhere else. Those seeds germinate, grow into an adult tree, and then slowly through time, those ranges change. Now, is anyone here familiar with the seed zone hardiness maps? Like what the USDA puts out to tell you where you can plant certain species in your garden, right? That kind of thing. The way that those maps are produced are through models that project what the future climate will be in a certain area and sort of map the species distribution onto that. So if we take a species like sugar maple, for instance, we can map what we think the climate will be like in the future, and that is essentially assumed to be where that species will exist in the future. Sugar maple will move upslope, it will move further north. There's a few problems with that general approach. One, if you actually look at those maps, they're at a scale that's at a continental scale, very big. It doesn't tell you what actually is happening at that local scale. And as anyone who's in forestry here will tell you, most management happens at a very fine scale, like at a stand level. So they may not be so useful in that way. The other thing is that they miss out on other things that plants respond to. They miss out on what soils are like, for instance. They don't take into account things, other organisms that these plants interact with. Pollinators could be missing from this. Herbivores could be missing from this. So picture, say, a sugar maple seed trying to grow beyond where it currently grows, let's say further north. The climate might be better. It might be warmer in the future for it to be able to grow. But we don't know if, say, a squirrel will come along and pick it off and kill it. We don't know if the soils are good enough to support germination of that seed. The other thing that is often missing from these models are any information about what's going on below ground. So that's where our fungi come in. So as a little example here, this isn't perfect. In any typical forest, you can imagine the biodiversity being everything in this glass. All the potential species that grow in a forest that you can find in a forest are in this glass. Everything that you see, all the trees, all the plants, all the mammals, the birds, could probably represent the head of that beard in terms of total number of species. Everything else is the actual beard part. You see? And that's not hyperbole, that's probably an underestimate. The unexplored diversity that is found in soils is mind-boggling. We could probably go outside right now with a shovel, dig a little hole, sequence the DNA of everything in there. We'd find at least 2 new species unknown to science. It's very stunning. And one of the things that makes up this species pool is fungi, in particular mycorrhizal fungi. Excuse me, I just need a beer. I'm going to drink some of the biodiversity now. Some definitions are really helpful here. Keep in mind, feel free to interrupt me with questions at any point. When I talk about mycorrhizal fungi, they're not exactly the same thing. And you might have heard of some of these things before. Let me try to break it down for you. Mycorrhiza is a symbiosis. A symbiosis means a relationship between 2 separate organisms. It doesn't imply what direction that relationship takes. There can be mutualistic symbiosis where both partners benefit. There could be a parasitism where one partner benefits to the expense of another partner. Of course, there's cases where one partner benefits and doesn't do anything to the other partner. Some examples of this could include a pollinator in a flower. An insect gets pollen, gets nectar, gets whatever, gets some sort of reward, the plant gets a reproductive service. That's a mutualism. Mycorrhiza, the interaction between a plant root and a mycorrhizal fungus that grows in the soil, in general, is mutualistic. There are certain cases where that's not true. Happy to tell you about those if time allows. But most of the time, it's a win-win for both parties. But what do they get out of it? What's the point of it? What they get is the surplus of resources from each partner. So let me just tell you what I mean by that. Plants are really, really good at photosynthesizing. They're really, really good at taking carbon from the atmosphere and converting them to sugars. The fungus, below ground, very, very good at foraging through the soil and collecting things like nitrogen, phosphorus, things that plants need. What each party is really bad at is what the other partner is really good at. Fungi are limited by sugar, by carbon. They don't really have good access to it by themselves. Plants, really, really bad at competing for nutrients below ground, like nitrogen and phosphorus. That's not always been the dominant thought, especially in forestry. When I was training as a forest ecologist, no one mentioned that mycorrhizae even existed. We now know that they aid most land plants in providing nitrogen, phosphorus, other soil nutrients that they can't otherwise get access to within their rhizosphere. Rhizosphere just means the soil around the plant root. Plants aren't so good at competing for that because other things that exist in the soil are better. Bacteria are much better at getting nitrogen and phosphorus than plant roots. So fungi kind of tip the balance. But what actually is going on between these two? Actually, it's really cool, and I wish I could show you pictures of some of these organisms. There's fossilized plant roots from 430 million years ago showing little tiny fungal structures inside fossilized plant root cells. Coincidentally, that's about the same time plants colonized land in the first place. Is this a coincidence? Probably not. Sorry, that's a bit loud. In general, the main takeaway is that in exchange for the carbon that the plants are able to easily photosynthesize and fix from the atmosphere, fungus provides nutrients. So that's like the general takeaway from that relationship. It's not the only benefit that these organisms provide each other. In recent studies, it seems that these fungi are also really good at redistributing water through the soil. In fact, in a lot of studies looking at the effects of drought on plants, ones colonized by certain mycorrhizal fungi are really good at resisting that drought, have greater survival. Further, there is a lot of evidence to show that these fungi provide protection from soil pathogens, from heavy metal toxicity. There's really cool studies that show that some of these plants that are colonized by fungi, I believe the example that comes to mind are some willow species, can actually grow on old mine tailings, super acidic, super toxic soils. They do okay. So there's a lot of nuance in this relationship. And what was really interesting to me when I first got into this was how this could relate to this understanding of where species exist. So how do these fungi, what role do they play in promoting seeding and establishment? How do they change where species grow? If climate change is really shifting where trees will grow in the future, what role do fungi play? And so it's helpful to understand sort of the basics of how these fungi actually exist in nature. I'm going to simplify it a little bit, but there are generally two main types of these fungi. There are muscular mycorrhizal fungi, which I will call AMF, and the ectomycorrhizal fungi, which I call EMF. AMF form little branch-like structures inside of the plant cell walls. They actually burrow into plant cell walls, and the resource exchange occurs in that space. It's important to remember that plant cells also have cell membranes. So imagine like a little plastic bag inside of a cardboard box. That's basically how a plant root cell works. In that EMF situation, ectomycorrhizae, ecto meaning outside, the fungal component exists outside of the plant cell walls, but they weasel their way in between the cells, and the resource exchange occurs in that space. With EMF, it's actually really cool. You can go find seedlings that are colonized by these fungi. You can see them with the naked eye. They form these massive sheaths over plant roots, and it's really amazing. There's a lot of diversity of colors, shapes, sizes. With AMF, it's a little trickier. You have to kind of use a microscope, but when you do see them, it's really striking. You will not likely find many root tips that are not colonized by some of these fungi. And it's interesting that we've only known about these interactions for a little over 100 years. The first person to describe these was a German forester in 1885. It's just that no one was really looking, which often is the case in ecology. Now, as it relates to where trees will migrate on the slopes of mountains, you have to remember that these fungi are somewhat specific in who they want to partner with. The AMF, as described, in this region, they colonize relatively few tree species. They colonize a lot of herbaceous species in the understory. A lot of your garden plants, for instance, may be associated with this type of fungi. Interestingly enough, they don't associate with things like broccoli or the cruciferous vegetables, for whatever reason. In the tree world, the AMF are associated with sugar maple, other maple species, really dominant deciduous hardwood species in this region. But the EMF, they're colonizing things like oaks, like birches, beech, pretty much everything else. As you go up in elevation and you encounter those evergreen species that we talked about, spruce, fir, those are all EM associating as well. So this was my question going into my work. If you're a sugar maple, you're trying to grow high elevation or high latitude forests, and your friends aren't around, do you do well? Do you survive? Can you even establish? Despite what all the models say, would you even be able to have a chance to grow in those places? So with that question in mind, I engaged in what some would call an overly ambitious study. So this was during my PhD. What I wanted to do was see what effect these partners had on the growth of tree species like sugar maple, and then EM trees like beech. It's a lot harder than it sounds. Namely because there's a lot of other things you have to keep in mind when you try to answer this question. One being, there are other things that correlate with changes in elevation. One being, it's much colder at the top of the mountain than at the bottom. You have to take that into account. The soils are very different. At high elevation, pH of some of the soils on these peaks can go down to 3. It's like orange juice. Almost nothing grows. Well, things grow, but not well. So how do you separate out these effects, the effects of these other factors, and focus just on the fungi? It's difficult. So what I decided to do is I decided to take soil from the field, bring it into a greenhouse. And I had this fancy machine called an autoclave that kills all living things inside of it once you put it in. So imagine me putting garbage bags full of soil into this machine, killing everything inside in one treatment. In another treatment, I just left it alone. Didn't do anything to the soil. Third treatment, I took some of the living biota from the live soil, put it in a potting mix. So what I've effectively done is separate out the effect of the soil chemistry from everything living in the soil, and then grew seedlings of various species in that soil environment, those soil environments. And what we find is that soil chemistry matters, If you are a seedling growing in this really acidic, nasty soil, you don't do very well. But independent of that, it seems that you need your appropriate partner to grow. If you were a sugar maple and there were no AMF around, you also didn't do very well. So that was the first thing I did. Second thing I did, same thing, but I took it to the field. I actually moved soils from different parts of the mountain to control the effects of climate on the growth of these seedlings. So imagine me, big 70-liter Osprey bag, carrying soil up the top of Mount Mansfield, down to the bottom of Mount Mansfield, at 3 different elevations. I did this for 4 mountains. It took a week, 100 pounds of soil at a time. No one helping me. It was the middle of the pandemic. Not complaining, I mean, it was fun in a weird, sick way. And I sowed seeds of sugar maple and beech, these various mycorrhizal types, into these soils. And like I expected, if you're growing at high elevation, you don't do very well. It's too cold, winters are harsh. But if you're a sugar maple seedling and you're growing with the soil from low elevation with all this nice AMF friends present, you do really well, regardless of where you're planted. I grew sugar maple near tree line on Mount Mansfield. And if anyone's hiked on Mount Mansfield, that's not really a place you find sugar maple. Partly because there's no appropriate mycorrhizal fungi present. So, through this study, it seemed clear to me that there was this role that was played by these fungi. They were helpful. They were aiding plants in establishing new places. And in my mind, this opened up a whole bunch of possibilities in terms of, importance in forest ecology, forest health, forestry, silviculture, possibly some implications for assisted migration. For those that don't know, there are people that think that taking the step of actually physically moving species to new locations will enable their survival long-term and their persistence. That effort may be a huge waste of time if you do not also consider the fact that these are species that require these mutualistic present. So it opened up a lot of doors for me. Yes?
So, a really basic question. I'm not sure I'm going to say this the right way. So
Yeah.
the fungi that you need,
Yeah.
I always think of it as being like connected with everything else. So can you, part of your study, was it like, could you take a batch of it and like stick it with the sugar maple and that was enough, or is it the interconnectivity that you need with the larger system to work?
That's a great question. So I was going to avoid that until the end. But I actually am going to transition to it now, so thank you for bringing it up. So what you're describing is, or at least what I think you're describing, is some aspect of what we call a common mycorrhizal network. And I'll just get into that right now. So, has anyone here read something called Finding the Mother Tree by Suzanne Simard? Maybe you've read Hidden Life of Trees. That's a more popular one. Yeah, a few of you. Fungi are having a glow up right now. They're going through a moment in our collective consciousness. You've probably also seen Fantastic Fungi, Paul Stamets' documentary. Yeah. So we've all suddenly come to the realization that fungi are incredible, beautiful, perfect organisms that will solve all the world's problems. Central to that belief is this idea of what's called the Wood Wide Web, or Common Mycorrhizal Networks, CMNs. What this is a theoretical connection between at least 2 plants via a fungal link, where the hyphae of a particular fungus is connecting multiple plants. And through that connection, the plants are able to share resources amongst each other. for instance, think the carbon or the sugars from one tree can go into the fungi, be translocated somewhere else and go to another plant. And a lot of people think that this is a perfect example of altruism in nature. I heard a laugh out there. It's a very compelling narrative. There's good evidence to suggest that it does indeed happen. It does occur in nature that carbon from one tree can go to another tree. Even sometimes the carbon from an adult or mature adult tree can go to a closer related seedling of the same species. What I'd like to try to, not convince you, what I'd like to share with you is maybe a little bit of a repudiation of that idea. It seems to me that that's a narrative maybe that's advanced a little far beyond the science. It's a very popular idea. I would love it to be true that that's the case. Unfortunately, the evidence for it is not very compelling currently. The studies that actually look at this, they use what's called radioactive isotopes. So they'll inject carbon-13, for instance, C13, into one plant and then trace where it goes. And if it ends up in another plant, they assume that that's evidence that there's this movement through the soil, through the fungi, to that tree. They very rarely take into account the fact that that carbon can just move through the soil without any fungi. It can be respirated from the plant, float around in the air, and be fixed over here by another plant. There are many pathways in which resources can move, and they don't really demonstrate that there's this movement through the hyphae of the fungi in particular. Now, I'm not generally a stickler on these things. Like, I understand the need for a good story, for us to care about forests in their own right. I would just say that, I don't think it's necessary to blow up a story and convince ourselves of something on bad evidence to support good action, to good forestry action, good policy, good environmentalism. The other thing that always bothered me with that narrative was that the fungi themselves have no agency in this whole phenomenon. The trees are simply directing the resources from one plant to the other. They're just telling the fungi to accept the flow. But the fungi need that carbon. That's their limiting resource. There's no real good reason why they would give it away. So I would advocate to you that we shouldn't think of fungi as just tubes in the ground that do the plant's bidding. And I used to think that. I was a plant-centrist guy. As I said, I was trained as a forest ecologist. I wasn't trained to think like a fungus. But they're organisms too. And the narrative also anthropomorphizes these organisms. The whole idea of this altruism, it's great in that it associates this, or ascribes this feeling that we as humans have to one another. But these organisms are really cool in their own right. I don't want them to be like people. I want them to be as they are. So I've always pushed back on that idea a little bit. But in terms of your question, I'm sorry I digressed a little bit. I did also conduct an experiment where I moved soil in the same way as in that field experiment. But I made these special pots that had these big holes cut out of them. And I stuck a mesh, a very, very fine mesh, that would allow the hyphae of those fungus through, but not plant roots or other things. In one treatment, I just left alone. In another treatment, I turned the pot 360 inside of another pot, breaking the connection that it might have had to the outside. If there are mycorrhizal network connections inside of that pot, they get broken. So it's essentially an experimental test of whether or not a potential network is having an effect on the seedling. Does that answer your question? Okay. So I can talk about the Wood Wide Web stuff a lot. I won't get into too much more, unless you have questions about it.
So where did you publish your studies?
Oh, what's that, sorry?
Where did you publish these studies? Are they still?
Yeah, so I recently published in ecological monographs. So it's all very easy to find. I can send anyone a link if they're curious. Any other questions? Yes.
When you were doing the experiment where you're moving soil from different locations on a mountain, did soil depth matter at all? Like, did you have a certain parameter, like how much, like how far down you were going?
Yeah, that's a good question. So if you didn't hear, the question was, did I take into account the depth of the soil that was moved? So they were all taken to a depth of about 30 centimeters, so into the mineral soil. They were all the soil I moved were taken at the same depth, so I didn't vary that. But it is a good question. So different fungi do exist at different depths in the soil profile. In fact, when I talk about fungi, there are many different kinds of fungi. Not all are mycorrhizal, not all engage in this behavior. Some are what we call sapotrophic fungi, which just means they're like the traditional fungi we learned about in elementary school. They're just decomposers. They're not just, they're decomposers. They don't engage in this relationship, and that's generally where we find those fungi is where there's the most accumulated organic material in the soil. So yeah, there's a difference in the profile. Yes?
So your implication is that the mycorrhiza, you can plant a sugar maple up high elevation because of the mycorrhizae, but it just seems to me, I'm sure you didn't stick around for 100 years to see how that maple tree is growing. To me, a huge difference between maple and balsam fir or spruce is the root systems and the other ones are very flat, two-dimensional, whereas the sugar maple is going to require a lot more depth of root mass. So it doesn't seem to me that, it's an interesting theory, but there's a lot of other factors there that are going to control whether that sugar maple could grow up there.
Oh, absolutely. And that's a fantastic point. So ecology in general is filled with these multiple hypotheses and these multiple potential explanations for what's happening. So for those who don't hear, the question was posed, is there a role for things like soil depth in facilitating the expansion of some of these species? And absolutely. For those who are not familiar, the soils at really high elevation are very thin. You can go in like sometimes even as much as a foot, but you'll hit bedrock. And some plants need as much substrate as possible. So absolutely, as a tree ages, maybe there's just not enough soil for it to cling onto. That being said, a tree still needs to get through that initial filter, that initial stress, to be able to get to adulthood. And it seems evident that fungi is one of those hurdles that needs to be overcome. Whether or not it'll survive to adulthood, another story entirely. And unfortunately, if there's any graduate students in the room, they know that all of your studies are going to be maximum 5 years. That's just how it goes. That's when the funding runs out or when you graduate. So there's a limitation in just the field in how we carry out science in general that contributes to our lack of understanding of some of these things. But my hope is that other people will carry on those experiments and find out more. Yes?
How many species, do you have an idea of how many species of active mycorrhizal fungi are involved with sugar maple? And do they exist in forest soils where sugar maple are not present?
Yeah, excellent question. So in the case of sugar maple, I have no idea how many fungal species they associate with. And the reason being is that AMF, the arbuscular mycorrhizal fungi, we know of about maybe 350 distinct taxa, distinct species. They're very cryptic. They're hard to sequence. They do not culture. You can't grow them. These are very, very challenging organisms to work with. I will say that as part of the next step of this research, we are actually going back, repeating some of these experiments. We're actually using molecular techniques to identify which species are involved in some of these interactions. That's sort of like the next stage of evolution of this work. The picture is a little more clear with the ectomycorrhizal fungi, the things that associate with beech, spruce fir. There's upwards of 50,000 species there, and they are represented by things you would identify if you're an amateur mycologist. A toadstool, amanita, the classic mushroom-looking thing out in the world. That's ectomycorrhizal fungi. Other tasty, edible mushrooms that you find, also ectomycorrhizal. But yeah, to answer your original question, we don't know. But we're going to try to find out. The other thing I want to note about that work, excuse me. The other thing I want to note about that work is the techniques that we use to identify species of fungi is very problematic. I'll give you an example. Be right. You're next. So in the traditional biological species concept, we define species as things that can't mate and produce viable offspring. So there's a clear differentiation between a human, a chimpanzee, gorilla, what have you. With microbial organisms, we use sequence similarity, because that's all we have to work with generally. And we use arbitrary cutoffs in sequence similarity to define what a species is. It's generally thought to be like 97% sequence similarity. If we apply the same criteria to primates, then us, chimpanzees, gorillas, gibbons, we'd all be the same species. You see the problem, but it's the best criteria that we really have. So applying unique species identifiers is challenging. You had a question.
Why is soil so acidic on the top of the mountain?
Ooh, that's a great question. Multiple reasons. One, it has to do a lot with the bedrock that underlays that material. And when it weathers, it becomes acidic just because the bedrock itself is acidic. The plants themselves actually have some like a feedback mechanism to maintain that acidity. So the spruce and fir trees themselves that are found at that high elevation, they have litter that itself is very acidic. These are things that have a lot of nasty compounds, lignin, tannins, things that don't break down easily. It's further compounded by the fact that since it's so cold up there, there's not a lot of turnover from the bacterial community. It doesn't really get broken down very quickly. So it kind of just accumulates and piles up. So if you just think of a soil that's really full of organic stuff, it's very thin. Yeah, it's maintained in that state over a period of time. I think it was you. Sorry.
You mentioned the fact that some of this stuff grows really well in a hostile environment, such as mining tailings and acidic environments. There's been a lot of effort in the last couple decades to work with bacteria, especially to break down landfills. And one of the big problems with landfills is when you dig in at any depth at all, you can pull stuff from 30 years ago, like a hot dog. There's steam, there's no oxygen down there.
Yeah.
There's heat, but there's no air. I'm wondering, how deep can these mycorrhizal that live in these hostile environments, how deep can they go, and can they be used for remediation? Can you tune them or select them to basically decompose?
Oh, wow, that's a great question. I have to think about that. As far as I'm aware, you generally don't find them super deep in the soil profile. They're generally organisms that operate well in an aerobic environment. Yeah, I'm not sure about that. That's a great question. I know there's been a lot of work to use fungi in remediation in general, not necessarily mycorrhizal fungi. That's more of like the Paul Stamets power of fungi kind of stuff. But yeah, that's good. I'm not so sure about that. Yes.
Well, this is more of an observation, but I recently, if you're familiar with the Appalachian mountain called Jordon Appalachia, I recently had an interview in there with George Schaller, who's a very famous wildlife biologist. And and Schaller is 90 years old. He lives in Lebanon. I was just shocked to find out he's my neighbor. And he had he's worked in tremendous, he's saying he'd been involved with conservation work like in areas like the sides of California in his lifetime. And he worked with a lot of big mammals, pandas, gorillas. And I asked him one day, I asked him about working with smaller or more nondescript animals. And he said, no one's ever going to pay me to do that or have any interest in that. So in that context, I'd like to applaud you for taking on something that just doesn't get a lot of attention or interest, and for bringing it forward in such a passionate way.
Well, I appreciate that. Thank you. I will say I did not get paid very much as a graduate student. So, I mean, there's that. But it helps to work at a nonprofit that values a diverse viewpoint and a diverse approach to research. So I have the luxury of working in an area that allows me to do that. But yeah, I appreciate that. Yeah.
Yeah, I was wondering, in where your studies, and maybe you don't know, but with your studies are more in the mountains and more undeveloped areas, but I'm wondering about in more developed areas, like whether it's a golf course or cemeteries or people's yards where they may have sugar maples growing or
Yeah.
a bunch of other native hardwoods, and how the fungi are there, and are they impacted by, say, if people are applying herbicides or tick stuff or any of the other host of things that get put down on lawns, and what do those do to the whole network?
Yeah, that's a great question. So I think it was you who someone approached me earlier about fungi in the context of land use legacy, how things have changed through time, forest clearing, that kind of thing. The real answer is there's a lot of active research into how communities of these fungi respond and change in terms of abundance composition of individual species through time, given those impacts. But really, we don't know how they respond to some of these things because we don't have really a baseline to compare to. We don't know what a community of these fungi in a more pristine or unmanaged state would have looked like. I mean, we can sort of glean clues from more intact ecosystems, but again, it's really hard. What doesn't help matters is there's such a wealth of diversity of fungi, and it's so heterogeneous. There's so much heterogeneity across the landscape. You could, again going back to that shovel example, you could dig a hole in one spot and sequence everything and get one unique suite of species, and dig a hole 2 feet next to it, and you might get a completely different community of fungi. It's hard to say. Almost certainly there is an impact of our activities on those organisms, but as yet we don't know to the extent that we are influencing them, and that's a major problem. Oh, I'm being told I have one more question. Yes.
There is a group of scientists who've been studying plant intelligence lately. Very exciting work. Monica Galliano comes to mind. Do you know anyone who's taken that work to the world of fungi?
Not by name. I know there is a big push to look into that work. I recommend a book by Merlin Sheldrake. What was the title? It was yes, Entangled Life. Thank you. That dives into that topic a little bit more. They definitely display some sort of quote-unquote intelligence. They're able to solve problems. They can reallocate resources through the fungal body in ways that would imply that they're responding to stimuli in a pretty sophisticated way. But yeah, that's not my field. Yeah. Yeah.
To what extent are these fungi dependent on other larger forms of life? Or is it possible, for example, that, say, on Mars we might discover them as the only form of life?
Oh, that's a psychedelic question. I go out on. Wow, that's amazing. Yeah. So I believe the question was the second part. Could it be possible that, say, on Mars, they'd be the only form of dominant life, right? Or the first part of that question, to what extent they're dependent on larger organisms here? Obviously, a lot of these fungi are obligate symbionts for these plants, so they need to associate. A lot of them need to associate with these trees. They're not exactly free living, so there's that. There's also evidence that things like herbivores have like an indirect role on their functioning. There's actually been cool studies that have shown that the dispersal of some of these fungi, the spores, it depended on small mammals like rodents. They'll dig around in the soil and carry spores around. In terms of the second part of your question, could it be a dominant form of life? Maybe. They're pretty sophisticated. But I don't know. That's a great question. All right, Jason's coming to relieve me.
Thank you very
Thank
much.
you so much. Thank you, folks.
Thank you, Jordan and I are going to hang out, and anyone is welcome to come and ask some more questions. Thank you all for being here tonight. Really appreciate that. Next month, we're meeting on Thursday, because I'll be counting votes at town meeting on Tuesday night. We're driving voting equipment down to be locked in the vault of the town building, so I won't be here. So don't you be here. I mean, you be here. That's great. And then come back Thursday for Suds and Science. Next month, we're talking about agroforestry. We're going to learn about how farmers right here in the Upper Valley are adapting to climate change, integrating trees and shrubs into their row crop and livestock productions to increase productivity and yield. That's next month. Thank you for being here, folks. We're going to hang out. Come say hello. Have a good night.